chapter 9 is entitled Hormones. You know, if it was up to me, I would also have entitled it Signal Transduction. Because really you'll see the hallmark feature of this chapter, which is a basis for pretty much any other disciplines afterwards, is how do I get cells to have specific responses when most of the chemicals are floating out there in the blood or are floating in a synapse and never having access inside that cell. Well, it's going to be thanks to binding to receptors. And what we want to study in this chapter is whether you're a hormone, a neurotransmitter, a toxin from a bacteria, a drug from pharmacology, wherever you're from, the whole idea is what is the sequence of event between the binding from outside a cell to the actual responses that occur because of chemical changes inside the actual cell. So as an introduction, the idea of hormones is that biochemistry, as you know, is going to be very much divided in two sections for metabolism. That under the control of insulin after a meal, and the portion that is under the control of glucagon, cortisol, epinephrine in between meals. When I'm looking at hormones and signal transduction, as it says on this first slide, hormones are any compound produced by a cell which, when binding to the receptor that it's supposed to, can change the metabolism of the cell, and that's providing it can actually respond to the hormone receptor complex. Most often, a hormone is made in one location, the receptors are found in other tissues. For paracrine hormones, they're secreted directly into the interstitium, have a very short half-life, and find their receptors very close to where they were secreted, basically on the cell that is next to them. Things such as prostaglandins or neurotransmitters are in some ways paracrine type of hormones. The hormones that float around in the blood are part of what we call the endocrine system, because endocrine, they are actually within our body, and literally they distribute, just like drugs, into any tissues. If they find the receptor, they'll give responses. The whole idea is that we have two types of chemistry out there. And the two types of chemistry to bring about here is, am I water soluble? In which case I can stay forever in the bloodstream or the interstitium, but I cannot get inside the cell, and I can't get into a cell. Why? Simply because cells have a phospholipid bilayer for a membrane. If I'm water soluble, I cannot cross the membrane. Conversely, if I'm a hormone that is lipid soluble, then I will be able to get into the cell, and if I can get into the cell, then I'll be able to directly affect the metabolism or the functions of that one cell. It is primarily the water-soluble hormones that we want to review here, because for the lipid-soluble one, you've already seen it in your molecular biology. You remember steroids? Steroids were binding intracellular receptors. And once the steroid was bound to its intracellular receptor, it would then allow the complex to go in the nucleus and bind to hormone response elements, also known as HRE. Recall that HREs are in the enhancer regions of spacer DNA. Is this a region that is expressed or not? I'm hoping you're remembering well that spacer DNA is never expressed. Enhancers will not be transcribed and therefore not translated in proteins. There are other places where when we bind to them, we would actually change the rate of gene expression. If we called enhancer, we would induce that gene expression, which, by the way, would that change the Vmax or the Km? If you enhance gene expression, do you make more copies of an enzyme? Or do you change the shape of existing enzyme? I know you're saying, well, if I induce gene expression, I make more enzymes. If I make more enzymes, I change the Vmax. Just a little link to your previous chapter. What we really want to study in this chapter is, therefore, how do water-soluble compounds manage to give responses? Their receptors are in the cell membrane. And the general pattern will be, once bound to the receptor, to activate what we refer to as second messenger systems. A key point of the second messenger system process is down the road to eventually turn on protein kinases. Protein kinases, they will phosphorylate protein. And phosphorylation of proteins can modify the activity of these proteins. We're in biochemistry, we're thinking enzymes as proteins. But if I was in physiology, I might think ion channels. If I was in molecular biology or genetics, I might think transcription factors. If I was in pharmacology, I might think other receptors might get phosphorylated or not. If I was in, I don't know, pathology, it might be actually extracellular matrix proteins or maybe contractile proteins such as myosin light chain. Whatever the case might be, protein phosphorylation changes the activity of that protein. I can't say if it'll turn it on or off. All I can say is that it will have a different activity. It is that process of going from receptor binding to this phosphorylation of protein that modifies the activity, for example, of an enzyme, that we want to study in the chapter. Well, as we take some examples of water-soluble hormones include insulin and glucagon. And let's remember that somehow these should oppose each other. So we're going to have to try and see if in the chapter we can explain how is it that at the molecular level insulin and glucagon should cause opposite responses. But you know, catecholamines, particularly epinephrine, will also be water soluble. In the lipid-soluble example, we have primarily steroids. It's also certain vitamins, such as the active form of vitamin D, calcitriol, thyroxine, the thyroid hormones, retinoic acid, a derivative of vitamin A. All these are examples of lipid-soluble compound. They will only work through control of gene expression. It may require hours, if not days, to do that job. Contrast insulin, glucagon, catecholamines. They may actually control the degree of phosphorylation that might require only minutes. But what you see on this slide is that with water-soluble hormones, you can also control gene expression. For instance, transcription factors such as CREB that you've reviewed in your molecular biology can respond to the levels of cyclic AMP, which will be a second messenger system, meaning there are two types of responses with water-soluble compound. Changes in gene expression that take forever to happen, but also changes in phosphorylation that may occur within seconds to minutes.